Maybe you've been hearing the term superbug. No, it's not some crazy evolved version of an insect like Ant-Man. When we talk about superbugs, many people are referring to this idea that some bacteria might evolve these traits which allow them to escape our antibiotics. So people might suggest, oh, because we're using hand sanitizers and we're using all of these antibiotics, our bacteria are evolving into these superbugs that are going to be really hard to destroy. Is there any credence to this idea? If so, what can we do about it? Is there anything we can do about it? In this video, we're going to take a look at bacteria and these superbugs. Let's get started. When we think about life, we might easily put life into two categories: eukaryotes and prokaryotes. Well, we know eukaryotic cells and eukaryotic organisms pretty well. Those are our protists, our fungi, our plants, our animals. What's less understood by most people are these ideas of prokaryotic cells or prokaryotic organisms, which may fall into what we call domain bacteria and domain archaea. Some people say archaea bacteria, but some people just use domain archaea. So what are these two things, and what are some of the characteristics? In this video, we're going to spend most of our time talking about domain bacteria because it's well understood. There is another domain, domain archaea. These are single-celled organisms that many people at first thought were grouped with bacteria. But what we actually understand about them now is that some of their metabolism and transcription translational controls are much more similar to eukaryotes. So domain archaea, while they may look like bacteria, are actually quite different. So in this video, what we want to do is we want to focus in really on the prokaryotes, specifically domain bacteria. So looking at the prokaryotic structure, let's take a look at some of the main details and characteristics. First of all, you'll notice it's unicellular. Their entire life is one cell. Their size, they're quite a bit smaller than our cells, than eukaryotic cells. At least 1/10 of the size, maybe smaller. And their internal structure, if you remember, prokaryotic cells do not have a nucleus or any membrane-bound organelles. They do have ribosomes, which you remember are for protein synthesis. However, the ribosomes that they have are a slightly different size than the ribosomes that we see in eukaryotic cells. So there is DNA, there is some genetic material in prokaryotes, but it is what we call circular naked DNA, meaning it is not wrapped around any proteins. It's just sort of loose, hanging out there in the nucleoid region. Prokaryotic cells also have cell walls. You may remember that plant cells and fungi cells have cell walls, not animal cells. But because these bacteria also have cell walls, it is one interesting way that we can help to combat them. So bacteria, prokaryotes, do have cell walls. They also have, as you see here, a plasma membrane as well. And then some prokaryotes move around, or they have these flagella, or singular flagellum, for locomotion or movement. So when you're thinking about the structure of a bacteria, the structure of prokaryote, they do have the things that all cells have: ribosomes, plasma membrane, cytosol, genetic material. But they also have cell wall, and some of them have flagellum. So let's think a little bit about the different sizes and shapes of bacteria. So bacteria come in many diverse shapes. One is cocci. And when you think of cocci, think of sort of this round sphere-looking structure. They also come in bacilli. When you think bacilli, think rod-shaped. Then you'll see some budding type bacteria, right? It looks like there's little buds. Then you can see some sort of spiral-shaped. You also can see some that look like a comma. So there's all kinds of shapes. But then we further classify beyond shape. We classify by how they're grouped. So the shape might be spherical, coccus, but if there are two of them, we might say it's diplococci. If there is a chain of them, we might say it's strepto, because strepto is chain. Sometimes you may have streptobacilli, so a chain of rod-shaped. Sometimes you might have streptococci, a chain of spherical-shaped. You might also see what we call sort of a grape-like cluster, and that is staph. I think of it as like a cluster, or look like a cluster of grapes. So you can see all the many ways we classify bacteria by their shape, by how they group, and then there are some more interesting shapes as well that are a little less common to see. So how do bacteria metabolize, or how do they get their energy? Well, some, believe it or not, get their energy from the sun. So think photo, light, troph, energy. So how are bacteria getting their energy? Some from the sun. And a good example of this is cyanobacteria. These bacteria here actually undergo photosynthesis. So they're getting their energy from the sun. Another interesting type of metabolism in bacteria are the lithotrophs. These are chemoautotrophs. And if you break that down, right, they're making their own energy from chemicals. So some of these inorganic compounds that they use would be like nitrogen, sulfur, hydrogen. They're actually able to get the energy out of these inorganic compounds like nitrogen, sulfur, and hydrogen. So a good example of this is thermodesulfobacteria. And you think thermo, you think heat, temperature, sulfo, sulfur. These are bacteria that can actually get some of their energy from sulfur. And then the most common when we think of bacteria that are around and on us are organotrophs. These are heterotrophs, meaning they get their food from others. So they have to live on plants and animals. So the bacteria that live on us, or in us that maybe cause diseases, or maybe that are beneficial for us, these are organotrophs. They're getting their energy from organic compounds, and we remember organic compounds contain carbon. Good example is Clostridium tetani, right? The bacteria that causes tetanus. So there are many different types of metabolism strategies for bacteria. The sun, inorganic compounds, and organic compounds. So when you do an experiment on bacteria, one of the hardest things to find out is, what is this bacteria, right? They're so small, they look the same. So if you have a bacterial infection, you go to your doctor and they're not sure what it is, they may take a swab and try to grow it on a petri dish. But even on a petri dish, it's hard to tell what bacteria it is. So we've developed some strategies for determining and grouping or classifying different bacteria. One of the ways we classify bacteria is gram-positive or gram-negative. And this is named after the scientist that had invented or discovered this technique. So some bacteria, as you'll note here, have a thick layer of a substance called peptidoglycan. And peptidoglycan is basically just some polysaccharides, all right, some sugars, and amino acid chains. Lipopolysaccharides are basically lipids and polysaccharides together, right? Lipopolysaccharides. So we find that in general, bacteria fall into one or two of these categories, gram-positive or gram-negative. The gram-positive bacteria have this thick wall of peptidoglycan on the outer part of their cell wall. Whereas gram-negative, they just have a small little bit of peptidoglycan, and they have these lipopolysaccharides on the outside. So what does this mean? Well, what it actually means is that they stain differently. So if we take some bacteria and we stain it with some different stains, gram-positive will look different than gram-negative. So this process is called gram staining. So what we do is we take the bacteria, we put it on a slide, and then we run a few different dyes or stains over it. We run a purple stain, and we also run sort of this pinkish, reddish stain on it as well. So when we run these stains, we put the stains on the bacteria, what we find is that the gram-positive bacteria will keep a purple stain. So when we're looking under the microscope and we see a lot of purple, we can say, this is the gram-positive category. Now I can further determine and do some more tests to see what bacteria it is. If the purple stain is no longer available, it sort of washes off, what we'll see is gram-negative. These appear sort of reddish or pink. So this immediately lets us know, is it gram-positive? Is it gram-negative? Now on this center diagram, you can see we've got some gram-positive, the purple. Also, we got some gram-negative. So if we wanted to isolate these and then further study the gram-negative, we'd know where to look. And if we wanted to do the same with the gram-positive, we could isolate them and then do some further studies or exploration on them. So this is really just a way for us to classify and help determine what kind of bacteria it is that we're dealing with. So we've talked about different types of bacteria. Now let's think about how they're genetically different. So bacteria have a lot of mutations. Well, that makes sense because a bacteria reproduces every 20 minutes. Imagine if humans were reproducing every 20 minutes, right? We would be overrun. Well, bacteria, because they're reproducing so fast, they show a lot more genetic variation. So how do bacteria reproduce? Well, it's pretty quick, every 20 minutes, and they undergo what we call binary fission. So basically, one cell becomes two. They basically just split in half. It's very similar to mitosis. So because they're splitting so much, because they are undergoing this binary fission, there are a lot of mutations because of the error rate in DNA copying. In fact, 1 in every 200 bacteria has a mutation from the original that it started with. So because they're reproducing so fast, the DNA has a lot of mutations in it. Sometimes those mutations are beneficial, sometimes they're harmful, and sometimes they're neither. They don't have any effect at all. So another way that bacteria have genetic variation is through what we call genetic recombination. So their genes recombine. So some bacteria may swap genes. And we did a lab on this. You may remember this was the bacterial transformation lab, where we put a small plasmid, which is just a little circle of DNA, into bacteria. So if here's our bacterial cell, there's its normal DNA there, right? And we might introduce a plasmid, a small circular piece of DNA that could be engineered, or it could come from another bacteria. And what will happen is the bacteria will treat that as its own. And it will replicate it, transcribe it, translate it to get what is needed from that particular piece of DNA. So how do bacteria have genetic variation? Well, 1, through their mutations. 2, through genetic recombination. So in this next slide, I want to talk about some more deeper sources of this variation. We just mentioned spontaneous mutation, right? Just DNA, because of the copies, so many copies being made, there's a big error rate. And then I just mentioned this idea of transformation, when a bacteria will take up a small plasmid, a circular piece of DNA, and then transcribe and translate it. And this was the lab we did with the piglo plasmid. We introduced this piglo plasmid, which made the bacteria glow in the dark, right? We introduced it, the bacteria took it up, and then some of the bacterial colonies actually glowed in the dark because they took in the plasmid that had the genes for glowing, right? Had the glowing genes. That's called transformation. The bacteria were transformed. They took new DNA and transformed the bacteria. Another source of variation is transduction. This is viral transmission. You may remember these little bacteriophages. Those are basically just little viruses, right? So there's the virus, and it injects viral DNA into the bacteria. So one way that the bacteria's genes can change is by getting some from a virus. That's called transduction. We also have conjugation. Some people say this is sort of like bacteria sex, where one bacteria is transferring genetic information to another and vice versa. Bacterial sex or conjugation. And then another one is transposition. This is just when segments of DNA move within and between other DNA. And we actually call these sort of transposons. Transposons. When a little segment of DNA, let's say it's that segment, moves from here to there. So the segment of DNA moved from one area of the DNA to another. So it was transposed from here to there. We would call that a transposon, and we call this process transposition. Okay? So there's lots of ways that bacteria show variety or can obtain variation: mutations, transformation, transduction from a virus, conjugation from another bacteria, or transposition within the DNA itself. So when we think of bacteria, often people are really worried, right? Bacteria are so harmful, they're bad, all bacteria are bad. And some of that is true, right? Some bacteria are pathogenic or disease-causing. So let's take a look at some of those to begin with. Now, bacteria can cause disease in humans and animals, but many people don't think bacteria can also cause disease in plants. So a lot of plant wilt and rot and blights are actually caused from microbes or small bacteria. And so here you can see a grape plant that's pretty much, all these grapes have pretty much been devoured, and even this leaf, because it has a bacterial infection, phytoplasma. So this phytoplasma infects the plant, uses the plant for nutrients, and then in turn sort of damages the plant. And then you think of bacteria often with all these diseases that we find in animals. So tooth decay, actually caused from bacteria on the teeth. Ulcers, right? Some of them are actually caused by a bacteria, Helicobacter pylori. And they invade your stomach cells and cause bacteria. So some people that have lots of stomach ulcers, that might be because they have a bacterial infection called Helicobacter pylori. Anthrax, botulism, right? These are bad words. You hear those and that's kind of scary, right? Botulism we can get from undercooked food. In fact, this is the reason we have to pressure can a lot of foods because we don't want botulism growing. And you can also, you've heard of botulism because that is used to make Botox. Plague, leprosy. What? Leprosy? Is that still around? Does that still happen? Actually, leprosy is caused by a bacteria, Mycobacterium leprae, also called Hansen's disease, and you can see some leprosy infections. That is bacterial. Several STDs or STIs, gonorrhea, chlamydia, those are bacterial. Tuberculosis, pneumonia, strep. Everyone in their life has probably had strep at some point, streptococcus. This, as you can see, here's a streptococcal pyogenes infection. And then also Lyme disease is caused by bacteria. Wait, Herder, I thought Lyme disease was caused by these ticks that come and bit you. That causes Lyme disease, right? Well, yes, but why? Because the the ticks are carrying a bacteria, Borrelia, which actually is what leads to Lyme disease. And you can see here in this example, the tick bit here, and we see this typical pattern that's around the bite mark. And this pattern is very typical in tick bites. So if you see this pattern, it's really a sign that you might want to get that checked out because you might have been bitten by a tick. So bacteria can be pathogenic, can cause disease. But as we'll talk about in a moment, these can actually most of them be cured pretty easily with some antibiotics because bacteria are living and an antibiotic would kill some living things in your body. So we talked about bad bacteria. Let's talk about some good bacteria. Bacteria, many of them are actually beneficial and necessary. If you think about decomposers, when we talk about the food chain and the food web, bacteria help to break down organic matter back so that it can be recycled into new organic matter. So this whole recycling of nutrients relies a lot on bacteria. Another important thing that bacteria do for us is that they fix nitrogen from the atmosphere. So if you think about plants, right? Plants grow, but they need nitrogen to grow, right? You think about all the organic compounds in your body that contain nitrogen. Well, we need some nitrogen. But the nitrogen in the air is really a form of nitrogen that the plant can't use. It's an unusable form. So what plants do is they have these bacteria on their roots, nitrogen-fixing bacteria, that literally fix the bacteria, fix the nitrogen from the atmosphere so that the bacteria can use the nitrogen to grow. So these bacteria we call nitrogen-fixing bacteria, and they are often found on root nodules. They are very important for nitrogen fixation. And then you may have heard about bacteria in terms of digestion. And this has become a sort of a really hot topic. It's this idea of your microbiome in your gut. And what these microscopic organisms or bacteria that are living in your gut do for you, all right? Well, a lot of them help in digestion. So think about cows, for example. Cows eat a lot of grass, right? Well, humans don't eat grass. Why is that? Well, we can't break down the cellulose. Well, how do cows break down the cellulose that's in this grass and these plants? They actually have beneficial bacteria that produce cellulase enzyme, which breaks down cellulose. So these herbivores that are eating a lot of cellulose, they rely on the bacteria in the gut to help them break down and get energy from this particular enzyme right there that's called cellulase. Also in our microbiome in humans, we're finding more and more that a lot of our vitamins and even some of our amino acids are produced by the gut bacteria in our microbiome. So bacteria have lived along with us for a long time. They've evolved with us, and many of them actually are very helpful to us. And we're even doing some pretty crazy things today with bacteria. We use bacteria to make foods for us, like yogurt. And we actually have been able to get bacteria, we've tricked it into making human insulin for us. So people that need insulin can actually get insulin today because bacteria, we've inserted a small little plasmid into the bacteria, and the bacteria thinks it's its own DNA, and it just makes a bunch of insulin for us. So bacteria can be beneficial and necessary. So let's wrap things up here with some cases where bacteria is not good, right? We talked about all those pathogenic bacteria, and we talked about, I briefly mentioned the idea, well, if you get any of those things, leprosy, strep, any of those bacterial diseases, you can take an antibiotic because antibio against life, since bacteria are living, we can take some antibiotics to help us get rid of those living cells. Well, the problem is this, if you think about it. If I'm taking a medicine that's going to kill living things, and I put it in my body, well, my body's got a lot of living things in there, right? I don't want to kill my cells. My cells are living. I don't want to kill them. So when we're designing antibiotics, or when scientists are thinking about antibiotics, we got to be careful and use some strategies to make antibiotics that kill the bacteria, but don't kill our cells that we need. And there's a few different ways that are a few different classes of antibiotics. If you think about it, one good way to kill bacteria is to kill their cell walls, because guess what? We, eukaryotic cells, we don't have animals, we don't have cell walls. So if we take a bacteria, or we take an antibiotic that kills the cell wall, it should just kill the bacteria and not our cells. Another interesting thing that we do to kill bacteria, many antibiotics target protein synthesis. That is, for bacteria to grow in your gut, right, they need to go from DNA to RNA to make the proteins to make more bacteria. Well, if we sort of try to cut this process here, bacteria can't make the proteins needed for survival. And if you remember way back, when we go from RNA to protein, we use ribosomes. Well, one of the interesting things about bacteria ribosomes is that they're a different size. So we can manufacture drugs that affect their ribosomes but don't affect our ribosomes. And there are several other ways we can try to attack the bacterial DNA. We can actually try to attack this pathway for folic acid. Folic acid is an important nutrient that bacteria really rely on, so we can actually try to attack this pathway as well. One of the newer ways to make antibiotics is by the signaling of antibiotics. And you may remember we talked about this idea of quorum sensing. And this is the idea that bacteria look around for each other before they start to attack. They want to get a quorum. And that all evolves around cell signaling. And so some of the interesting new antibiotics that I think are going to be coming out involve Dr. Bonnie Bassler at Princeton and her work with this quorum sensing and cell signaling. The idea that we can specify an antibiotic to kill only particular bacteria, the ones we want to kill, and not kill all the good ones in our body. So that's a really interesting new development in antibiotics I think we're going to see more and more of. So we started this video with this idea of superbugs and resistance. And sure, if you look here, we can test antibiotics and see how well they work. This is a typical antibiotic test where you take a particular antibiotic, you put it right there. Let's say it's methicillin or penicillin. You put it on this disc, and you see how far the bacteria stay away from growing, right? So this bacteria looks like this one's pretty strong. The bacteria are not getting anywhere close to it. Maybe this is a different concentration of that antibiotic. But the interesting thing here is that even when we have these antibiotics, like penicillin, methicillin, etc., there's going to be some bacteria, if you look really closely, that survive, right? So some of those bacteria evolve. They have the ability to be resistant to our antibiotic. And this is actually becoming a problem because as we give more and more antibiotics, as we're using more and more hand sanitizer to keep ourselves safe, some of these bacteria are evolving to be resistant to our antibiotics and cleaning supplies. One you hear about a lot, MRSA, methicillin-resistant Staphylococcus aureus. So it's this bacteria here, Staphylococcus, but it's become resistant to methicillin and some other antibiotics. So if you get a MRSA infection, it can be pretty damaging because it doesn't respond to a lot of our antibiotics. It's become resistant to it. So we have to continue to develop more and more antibiotics to fight against these infections. So if we go back, are we creating superbugs? Well, yes and no. We certainly are using antibiotics. We're certainly, especially in pandemics, using a lot of hand sanitizers. And those can affect evolution of bacteria. But it's a catch-22, right? We don't want to be sick from these bacteria, but we also don't want them to evolve really fast and become resistant to our antibiotics. So what do? Well, I think one of the best answers is moderation in everything, right? Don't take antibiotics if you don't need them. Don't take an antibiotic if you have a viral infection because the antibiotic doesn't kill a virus. So superbugs, are they going to be around? Probably. But hopefully science will continue to evolve more and more antibiotics to win the ever-waged war on us against bacteria.